Hello my dear students, here I am once again in front of you with yet another topic of biology which is interesting as well as confusing. It's a source of great trouble to many students. Today we shall try to bust that, okay? So the topic is pedigree analysis. Pedigree analysis is a method by which we can evaluate the chances of offspring bearing a particular genetic trait if that is prevalent in the family. Okay, pedigree analysis. It is done in case of Mendelian inheritance. Mendelian inheritance, as you all know, it is the inheritance of those characters which are controlled by one gene which is present in one chromosome. As against this, there are many factors, many traits which are polygenic in nature, polygenic inheritance. These are controlled by a number of genes, and the chances of their falling in children cannot be estimated because it is just so widespread. We cannot study those diseases, inheritance, like diabetes or blood pressure, high blood pressure, low blood pressure. Many lifestyle diseases are there. Then there's arthritis, so on and so forth. These diseases cannot be studied under pedigree analysis because they are simply not many chances of their having, and it's also it happens among many people. Okay, it's not rare. So here we are talking about some rare trait like as has been already found out, hemophilia, colorblindness, then Parkinson's disease, thalassemia. These are not so much widespread in society, but as with population increase, they are also becoming quite prevalent. But then still, we can find out the chances of their coming in offspring if parents are having, or if parents are having. If it is prevalent in the family, then we can find out the chances of offspring bearing such trait. So, pedigree analysis. Suppose there is a marriage in a family, okay, and the girl, or say girl's father, is albino. Albino is yet another genetic disorder, okay, albino. Now, in the male side, in the boy's side, not in immediate generation, anybody is known to be albino. Okay, immediate generation means boy's father or father's father or anybody in the family, they are not albino. But girl's father is albino. So, what are the chances of their offspring being albino? So, I am talking about those traits which are controlled by one gene. These are known as Mendelian traits. Okay, for Mendelian disorders, pedigree analysis is workable. So we shall just try to understand what this is and also what are the requirements to understand pedigree analysis. So come children, let's start. Yes students, so as I was talking about the requirements for understanding pedigree analysis. First requirement is to know the disease, okay, where it is prevalent, where is this gene located. So let us see. So as we all know, we have 46 chromosomes in our body, of which in every cell of our body, of which 44 chromosomes are autosomes, and 2 chromosomes are sex chromosomes. Now, if we are talking about this gene being present in chromosome, it can be present in any chromosome, locus of any chromosome, isn't it? It can be autosome also, it can be sex chromosome also. Now, if it is autosome, it can be dominant. As we all know from Mendelian inheritance, dominant means those characters which are expressed, recessive which are not expressed, which are hidden, and can be expressed only when they are present in homozygous condition. So, the disease, it can be dominant, that means it is it will show its character over the other gene, okay. 2 genes are needed, 2 alleles are needed to express a character. So, if it is present, it will express, okay, even if the other one is similar or some other. This is dominant, it will express. So, example of autosomal dominant is tongue rolling. I don't know, I haven't seen anybody in real life, but then it is there in our practical books, so we know of this particular disease. So, I mentioned. Then there is Huntington's disease. Okay, Huntington disease. Name is very musical. So, you can keep this disease in mind. It might come in short questions sometimes. Then there are recessive disease. Recessive, which as you know, they will be expressed only when both the genes are available. Okay, both recessive genes are together in a chromosome. Only then recessive can be expressed. Poor fellow. Example is albinism, as I was talking about, albinism, then sickle cell anemia. These are all autosomal recessive disease. PKU, phenylketonuria, autosomal recessive disease. Then we have sex chromosome. In sex chromosome, we have X chromosome and Y chromosome. X chromosome, again, similarly, it can be dominant recessive if you know what is dominant and recessive. So, if it is dominant, then only one X chromosome is needed to express its character. I mean, one X chromosome means only one, because the other might not be having this defect, but still, if it is present in one chromosome also, it will show, it will express. That is what dominant means, isn't it? So, example is rickets. Okay, so this is one common thing, name you should know because short questions again. MCQs, they are very probable chances of MCQs coming from these this topic. Okay, then X chromosome recessive. This is very common. Hemophilia colorblindness. Hemophilia, as you must be knowing, it is known as bleeder's disease, and it was first found out in Queen Victoria, and it spread among many members of her family, and many members of her family died because of this disease. It's a lethal disease. Okay, hemophilia. So that's why it became famous, because it was found in the royal family. Okay. Then color blindness is there. So these two diseases are recessive X chromosome diseases. Now, one thing you should keep in mind, X chromosome disease found in X chromosome locus, they are known as sex-linked. Now with sex-linked, don't confuse yourself with Y chromosome. No, Y chromosome disease are not known as sex-linked. X chromosome disease are only known as sex-linked. So keep this in mind. Y chromosome disease, they are known as genes. Y chromosome genes, they are known as holandic genes. Okay, holandic genes. So if question comes, what are holandic genes? So these are genes located in Y chromosome. So this is first requirement that we need to know what are the types of genetic disorder and that may be for which we need to do pedigree analysis. So they have to be Mendelian inheritance. This is very important. They have to be Mendelian inheritance. There are other genetic diseases, as I told you, polygenic, and yet another disease which are controlled by a particular chromosome. Okay, like increase or decrease in the number of chromosomes, not genes. The entire chromosome may be either double. Suppose we have two chromosomes for all the chromosomes, they occur in pair in case of somatic cell, isn't it? But if suppose instead of pair, a particular chromosome is found in three, okay, threesome, or it is only one of the pairs is missing. So these are the conditions for genetic disease like Down syndrome, Turner syndrome, Klinefelter syndrome. Those are because of either chromosome present in more number or it is absent. One of the chromosome is less. So those are not Mendelian inheritance. Okay, Mendelian inheritance are those diseases which are controlled by one gene present in one single locus of the chromosome. So these are those genes, and we can study pedigree analysis in these cases. Okay, this is the first requirement. Now I'll talk to you about the second requirement. So come. Yes, my dear student, the second requirement that we need is to know the symbols, because pedigree analysis is just a combination of symbols, isn't it? So let us try to understand what the symbols mean. Square means male, clear square means male, clear circle means female, shaded square is affected male, shaded circle is affected female, half shaded circle is carrier. Then square or circle which is crossed or rather just given a line like this, it means they have died. Then a symbol like this in which there is a female and male and a line is there, marriage. Then this kind of symbol, it means twins. Now this can be two boys also, two girls also, boys or a boy or a girl also. This is non-identical twin or fraternal twins. Then this second picture, this is of identical twins. Okay, there will be a line in between, and they can be either two boys or two girls. Now this picture is very important. You can see a shaded square or shaded circle, and there is an arrow beneath them, isn't it? This means they are propositors or proband. These are the first individual who have gone for treatment. Okay, or treatment has been taken seriously in case of these people. They are gone for study, analysis, all these things will be done in them. Their predecessor, they passed on with the disease just like that. They didn't go for any therapy. But these people, they are going. Quite responsible people, because after all it's the question of their offspring, isn't it? So this is what we need to know, pedigree analysis. Now let us move forward and see case studies. We shall try to understand pedigree analysis with the help of case study, and I can assure you it's very interesting. Okay, so come, let's move forward. This is our first case history. So let us study this. So when there is a analysis, then there has to be an observation and inference. Okay, so let's mark our observations. Observation, from this analysis we can see that first generation, this is generation 1, generation 2, generation 3. Generation 1, generation 2, generation 3. Okay, so first generation, they are not affected. Second generation is affected. Third generation is not affected. So observation 1 is skip of generation. First observation is skip of generation. Okay, now inference from skip of generation will be that it is a recessive trait. Inference will be recessive, because skip of generation otherwise will not be seen. If it is dominant trait, then it will fall, it will come in every generation, because it is there by its, if in one chromosome also it is present, then it is going to express itself. So the chances are too bright for it to be expressed in every generation. Okay, so there is no skip of generation in case of dominant trait. That is seen only in case of recessive trait. So we are seeing skip of generation, inference will be it is a recessive trait. Then second observation is, look at generation 2, there are 4 individuals, 4 offspring, of which 50% of female, 50% of male, they are affected. That means it is coming in equal chance in both male and female. So observation 2 is equally found in male or female. Equally found in male or female. So the inference from this will be, if it is equally prevalent in both male and female, that means it is not controlled by sex chromosome. It is autosomal. So our inference is this particular trait, it is autosomal recessive. Inference, the disease is autosomal recessive. Now, with further studies, it can be found out what are the chances of their offspring carrying this disease. So, this is how pedigree analysis is done. Here, the point that is to be noted is that if it is prevalent in both male and female, in boy or girl, brother, sister, then it is autosomal in nature. Okay, and if there is a skip in generation, that means the disease is recessive. So these are the points which we have to mark. So this is first pedigree analysis. Let us proceed to the second one. Second pedigree. Here, let us observe it first. Father is affected, then one of the daughter and son is affected. Affected daughter is married to normal man, and among their children, one boy and one girl are affected. So, observation. What will be our observation? Let us see. Is there any skip of generation? Skip of generation? No. Because it is coming in all the generations. So, there is no skip of generation. Inference will be dominant trait. Then, the second observation that we have to do is it occurring only in boy or girl? Here it is boy affected. Here one boy, one girl is affected. Here one boy, one girl is affected. That means it is coming in both male and female sexes in normal ratio. So equally present in boy or girl. So our inference will be it is autosomal or sex chromosome related. It is autosomal. Dominant. This particular trait is autosomal dominant in nature. Okay. Dominant because it is coming in all the generation. There is no skip of generation. And autosomal because it is showing in both male and female in equal ratio. So our inference will be this particular trait, it is dominant autosomal. Let us go through the third case history. Pedigree 3. Female and male. Male seems to be affected of first generation. Second generation, they are all fine. No one seems to carry the trait. Third generation, say this girl is married to a boy. Third generation, again the male member, one of the male members is seen to carry this trait. So what is our study? What is our observation? First point, is there a skip of generation? Yes, there is distinct skip of generation. Is it equally present in male and female? No, it is not equally present in boy or girl. Only the boys seem to have this. Okay, like grandfather had, then father's generation was all fine, then the son had. Now this son is about to be married maybe, so he is proband. He has gone for analysis. Okay, so this is proband. He is responsible citizen. He has gone for analysis, so proband. So our inference will be, this is first of all sex linked. Okay, this gene is present in X chromosome. Why X chromosome? You can ask question. It can also be present in Y chromosome. Why only X chromosome? The reason is, had it been present in Y chromosome, then all the generations would have, the boys of all generations would have got this, because Y gene is passed on to the boys. Okay, so even generation 2 would have had this disease, but they don't have. So this is X chromosome. Okay, there is a skip in generation, so it means it is X chromosome. Then secondly, is it dominant or recessive? Again, it is recessive because there is a skip of generation, so it is recessive. So our inference is this disease is sex-linked recessive disease. Now, this is the interesting disease that I was talking about, Queen Victoria. Okay, example of such disease is Hemophilia Colorblindness. So Queen Victoria had this disease, and since she was female, she had 2 X chromosome, and this X chromosome is recessive. She had a dominant, a good X chromosome which masked this character. She did not have any problem. Okay, she went on to live a very long life, but her sons, many of her sons died because since male member, they do not have another X to support it. Okay, so if it is present in one number also, then that disease will show, because the counterpart is Y chromosome. They do not have any gene to counteract the effect of this gene. So the chance of falling in them is very high. Their expressing the character is very high. So the boys are the ones which bear the brunt of this disease. Now here, this female who was married to a normal man, she was a carrier. But my dear students, this will not be given in your question paper if you happen to get such kind of question. Carriers are not shown, because otherwise it becomes very easy. Okay, she is a carrier, and she has passed it to her son. So this particular inheritance is also known as crisscross inheritance, because from poor father, he had 1 X chromosome which was defective. He has passed it to his daughter. All daughters will get X chromosome from father. Now, she had a normal X chromosome coming from mother, so she did not have any problem. She went on as carrier. Queen Victoria was a carrier. Okay, so she went along her life as a carrier. It never showed up in her, but nevertheless she had that X chromosome, isn't it? So she passed 1 X chromosome to her son, which was defective. And since he did not have any counter X chromosome with him, because he was getting Y chromosome from his father, so he had nothing to protect him. And this poor fellow, he was diagnosed with this disease. And many of the members in her family, they were deceased because of this particular trait. Because in this disease, if there is even a minor cut, it goes on bleeding and bleeding. So that's what. So this is our case study, and this is our analysis. This is sex-linked recessive disease. Now let's see the 4th analysis. Here we have, in generation 1, the female is affected. In generation 2, one female and one male is affected. That means one of its sons and one daughter is affected. And when the affected son is marrying a normal girl, we see that this trait is again showing in the females of the third generation. So, what will be our observation? Let us go through that. Skip of generation? Clearly no, there is no skip of generation. So, since there is no skip of generation, the trait will be dominant. Now, is it equally showing in boy or girl? Let's see. 1, 2, 3, 4. In 3 generations, 4 girls, 4 females are affected. But as for boy, only 1 boy is affected. So, it is not equally present in boy or girl. So, clearly it is X-linked dominant trait. Okay. This disease is present in X chromosome and it is dominant. That is why this female is having 2 X chromosomes, of which one is dominant and one is recessive. So, dominant is showing this trait. She passes dominant to 2 children, recessive to 2 children. Okay. So, the one who have recessive genes, they are normal, and the dominant, both son and daughter, they will get the disease. Even if this one is getting a good gene from father, but then the disease which is carrying, the gene which is carrying disease is dominant, so it will mask the recessive character. Then, now the affected son is marrying a normal girl, and since father will pass his X chromosome 100% to the girls, and that is a dominant trait, so all the girls of third generation will be affected. So, this is a sex-linked dominant trait. So, this is pedigree analysis 5. Let us see what we see here. First generation, male is showing. Second generation, again one male is showing. Third generation, 2 males are showing. That means, is there skip of generation? No, it is present in all the generations. Generation 1, 2, 3, all are showing this trait. Is it present equally in boy or girl? No. Clearly we can see that none of the girls are affected, only the boys are affected. So, it's a very simple deduction that this is present in holandic gene. That means present only in case of Y gene. It is present only in Y gene. So, this is Y gene carried disease. This disease is present in the gene for this disease is present in Y chromosome. So, my dear student, this is how we do pedigree analysis. Whenever we get to see a trait, we go through 2-3 generations back, and then we try to evaluate if this disease was prevalent in any other member of the family and how is this passed on in the next generation. From this, we can know for ourselves that will it be present in subsequent generations or not. So, this is all for today, my dear students. I hope you have understood the concept. I would suggest you please go through the case studies again and again so that you will get the idea of how to do this analysis. There is nothing, just you have to keep 2 points in mind. One is skip of generation is seen or not, and the other is it present equally in both boy or girl. And if you just get hold of this 2 facts, then pedigree analysis is a cakewalk for you. Okay, and these kind of questions, they are mostly present in question paper, and it's very easy to score marks here. So please go through this video and try to clear the concept. That's all for now, till we meet in the next video with yet another interesting topic. Bye bye and take care of yourself.